All right, so welcome everybody. We're going to get started. I'm Daniel Haber. I'm the director of the Cancer Center at Mass General Hospital, and I get to be the host for this week's Connect Science. So welcome to DFACC Connect Science. You can see the website listed here, and feel free to check this out, both if you want to look at upcoming talks. These are weekly seminar series, as well as recordings of past talks, or even a recording of this talk today. The guidelines, please, you're welcome to listen to the talk. Please type in your questions in the Q&A, not the chat box, but in the Q&A. And at the end of Leif's talk, we'll go through as many questions as we can. And then any questions that we can't answer at the end of this lecture will be emailed to Leif, and he'll have a chance to answer you directly by email. So today's seminar speaker is Dr. Leif Ellison, who's the Director of Breast Medical Oncology at the Mass General Cancer Center and Professor of Medicine at Harvard Medical School. He's an authority on breast cancer genetics, and his title today, as you can see, is Unanticipated Vulnerabilities of Chemo-Resistant Triple-Negative Breast Cancer. Leif, it's all yours.
Thank you, Daniel. Thank you for the introduction, and thank you for the invitation to speak. It's really a pleasure to address everybody today. What I'd like to talk about over the next 45 or 50 minutes or so are kind of some of the basics of our understanding of triple-negative breast cancer as the most refractory breast cancer subset, how we think about it both in terms of intertumoral heterogeneity and intratumoral heterogeneity, and how that is leading us to thinking about new ways we might subset and ultimately more rationally give treatment to patients with this disease. So first, a few slides of introduction. Get it to advance. Let's see, I'm trying to go. So traditionally, triple-negative breast cancer has been classified in a number of different ways, one of which was through gene expression profiling, where unsupervised clustering analysis has led to a number of different types of TNBC being defined, basically, as you see here, in a heat map of different gene expression profiles, where subsets are defined by largely the epithelial character and whether the cells were more basal-like, luminal-like, or mesenchymal-like, and then also how much immune stromal component is in the tumor. And this has led to a classification called the TNBC type or Lehman classification, which tells us something about the biology of the tumor. On the right, you can see a number of related classification systems based on gene expression profiles that are overlapping. However, this way of dividing triple-negative breast cancer has not been particularly useful in terms of prognosis for patients. And furthermore, it has not been particularly useful in terms of guiding therapy for patients, but nonetheless is somewhat of a useful way of dividing up the general biology of triple-negative and speaks to the heterogeneity between different patients as regard to their tumors. Another way to look at triple-negative breast cancer is through its fundamental pathogenesis. And fundamentally, triple-negative breast cancer is a disease of failed DNA repair. That's most clearly illustrated by the fact that germline mutations in DNA repair genes, particularly those associated with homologous recombination-mediated DNA repair, predispose to triple-negative breast cancer. This is a study of germline mutations associated with triple-negative breast cancer published a few years ago, and each dot tells you the mutations that have been associated with triple-negative breast cancer in various cohorts. So the big players in terms of germline mutation in triple-negative breast cancer are, of course, BRCA1 and BRCA2, as well as PALB2, but also other repair genes, and again, particularly those associated with homologous recombination-mediated repair. This understanding has led to very useful advance in therapy, which is the application of PARP inhibitors with those with germline mutations in HR-associated genes such as BRCA1 and triple-negative breast cancers. PARP inhibitors are both more effective and better tolerated than chemotherapy, so this is a significant advance, but unfortunately, as you see here in the upper left, This only applies to a subset of patients, maybe 10% or more patients who have germline mutations, and an even smaller subset who possess somatic mutations in these particular genes. So for the large majority of triple-negative breast cancers, this understanding has not led to new therapeutic advances. Another way to look at intertumoral heterogeneity of triple-negative breast cancer is through the prism of how these DNA repair genes get inactivated. And this slide summarizes some of the findings of a study that we participated in with Paz Pollock and Gad Getz a couple of years ago, looking at the mechanisms by which HR genes were inactivated in triple-negative breast cancers. And one of the striking findings was that the mechanism of gene activation in triple-negative actually varied by race. So what the chart shows is the prevalence of the different kinds of inactivation events. That's the vertical bars. And on the right, you can see in Caucasian patients with triple-negative breast cancer, the purple bar here shows pathogenic germline mutations. Next to it are somatic mutations, and as I mentioned, pathogenic germline mutations are about twice as common as somatic mutations in these genes. And if you put these together, the prevalence of those is substantially greater than epigenetic inactivation of these same genes in Caucasian triple-negative breast cancer. However, quite remarkably, and this was a complete surprise, that in African Americans with triple-negative breast cancer, epigenetic inactivation is far more common than genetic or somatic or germline inactivation. One of the reasons this is important, other than speaking to some interesting biology, is that currently in the clinic, we only screen for the genetic events in terms of which patients we stratify for PARP inhibitor therapy. We never use clinically the epigenetic silencing, and yet this study suggests that it's going to be quite important in directing the right patients to a better and potentially more effective therapy. So that tells you a little bit about the diverse nature and intertumoral heterogeneity in triple-negative breast cancer. But also important and a driver of the biology of this disease is intratumoral genomic heterogeneity, also referred to as subclonal heterogeneity. And this was alluded to a number of years ago, even before we were doing a lot of single-cell gene expression and other analyses of these tumors. And this is a study from a Nature paper in 2012 where they inferred the subclonal or intertumoral variation within triple-negative breast cancer simply by looking at the distribution of allelic frequencies of mutations across the genome from your basic exome sequencing study. So the different allelic frequencies could give you clues to the inferred subclonal diversity of the tumors. And what you could see on the right is that triple-negative breast cancers that had a basal-like gene expression profile, which are generally more aggressive and worse prognosis, had substantially more subclonal or intratumoral diversity, that is, more subclones that were distinct genetically within the tumor than did non-basal better prognosis TNBCs. And so in studies in the future, we're now beginning to look at what the nature of that diversity is and how it might be important in telling us about outcomes in triple-negative breast cancer. We do know, however, that clinically this heterogeneity and the subpopulations within the cells are likely to be the key toward increasing cure rates for triple-negative breast cancer. And this comes from a very fundamental clinical observation in this disease, which essentially is that if you take patients with triple-negative breast cancer confined to the breast who could be cured, and you give them chemotherapy, which is always given for triple-negative breast cancer, preoperatively, and then you perform surgery, in patients who have a complete response in the breast, that is, all viable tumor is eliminated, that's called a pathologic complete response rate, or PCR. Patients with PCR have an excellent survival, and this is a chart that summarizes this for a couple large cohorts. Their survival long-term is near 90% or more for modern chemotherapy in a patient who achieves a pathologic complete response rate. In stark contrast, though, an incomplete response, and that even could be just a few viable tumor cells within the tumor. Just about anything of residual tumor within the residual tumor after preoperative chemotherapy at the time of surgery is associated with quite poor survival and long-term survival rates around 60%. So think about that. I mean, what it tells you is that despite the fact that you've killed off the vast majority of the tumor cells, that really what's driving the long-term outcome of the patient is the subpopulation of tumor cells that remain and did not respond to therapy. So this is really a launching point for, number one, how we treat patients to try to understand these populations, and also speaks to the need to analyze these subpopulations in the tumors. So what I want to tell you about then with that introduction is really two interrelated stories. One having to do with single cell analysis of primary triple negative breast cancer, where we're trying to define these subpopulations within the tumor that might be shared across patients and understand their biology. And then secondly, about what we can do about the subset of triple negative breast cancers that are particularly chemo-refractory. What indeed might they respond to, and why are they chemo-refractory? So the first study I'm going to spend a bit less time on since it's already published. This was published in a really terrific collaboration between Miraban Karaivaz, a very talented instructor in the lab, and our computational biology collaborators, Simona Cristia and Francisca Mayor at the Farber, and Michelle Specht, a leader in our surgical oncology group here at Mass General. And so this was a very simple study in concept where we took primary triple negative breast cancer, dissociated single cells, and sorted and performed single cell RNA sequencing. So this slide summarizes the cell counts and general cell identities that we found in the first cohort of 6 patients that we published with this analysis. This was an analysis of about 1,500 cells totally, which, by today's standards is quite small, but this was a couple years ago. Here you see the 6 patients and the individual cells as columns in the heat map, and then the rows are individual genes that we selected to define the specific subpopulations within the tumors. What we realized as soon as we began sorting these tumors was that in some of the tumors, such as patient 84, more than half the cells were actually immune cells. And in this study, we really were interested initially in looking at tumor cells, and so subsequently we depleted immune cells from a number of the tumors that we analyzed in order to further enrich for tumor cells. However, you can see that even outside of the substantial immune components, there's a remarkable variation in the distribution of different subpopulations of different cell types within the tumor, such as patient 39, who had a substantial stromal component, and patient 81, who had essentially a 0 stromal component within the tumor. So remarkable diversity there in the subtypes. Now, what if we look at the individual subtypes of cells? These, of course, are t-SNE plots where we look in an unsupervised way at the different individual cells and ask what the different cell subsets and cell types have in common. And we found, as have others in the context of other tumor types, that if you look first on the left at non-tumor cells, that by and large, non-tumor cells cluster in an unsupervised way by cell type. For example, in the lower left, macrophages in dark green, and the upper right, stromal cells here in light blue, clustering by cell type. In contrast, on the right, you see tumor epithelial cells where the most prominent clustering is indeed by patient. So the colors here are individual patients, and you can see the clustering largely by patient, although of course some patients have multiple distinct subclusters. Now, of course, one of the big questions in all these analyses are how do you define tumor cells versus non-tumor cells? And we did that in a variety of ways having to do with genomic analysis, inferred genomic variation in subpopulations, and intratumoral heterogeneity to assign which of the epithelial cells were the tumor epithelial cells. And for brevity, I'm not going to go into all these details. But suffice it to say that having identified tumor epithelial cells, of course, we then wanted to ask about different subpopulations within individual tumors. And specifically, the hypothesis that perhaps there were individual subpopulations that had similarities across patients that might tell us something about the biology and the behavior of the individual tumors. And so what Simona then did was do a regression analysis to account for patient-specific differences and then look to see the degree to which she could identify subpopulations of tumor cells that were shared across patients. And the upshot of that analysis is shown here, again, in another Tisney plot, where the color coding represents 5 distinct clusters based on the analysis that are 5 clusters of types of subpopulations that are shared to various degrees across all patients. Of course, a 2-dimensional representation of a Tisney plot doesn't tell you exactly how these are interrelated, and so you might not say that this clearly shows 5 clusters, but if you looked at it in the multidimensional way that we conceive of Tisney, then they really are 5 subpopulations. So there are indeed shared subpopulations with common features, and I'll draw your attention to this green subpopulation, which we call cluster 2, that I'll have more to say about. So the first thing we wanted to know is, of course, what can we say about these subpopulations? So we applied to the subpopulation these known gene expression signatures, both expression signatures reflecting normal epithelial cells in the breast and signatures of the TNBC types that I have alluded to earlier. And a summary of that data is shown in this slide. So again, what we have here is, as columns, we have the patients and the individual cells. At the top, we have, in an unsupervised way, the individual cells based on their cluster, color-coded as in the same way that they were in the prior slide. And then the rows below are the individual gene expression signatures that we interrogated. So, for example, you can see that the patients with cluster 3, this green cluster here, patients 84 and 89 in particular, these cluster of cells resemble to some degree mature luminal cells of the normal epithelium that also shared properties with the luminal-like triple-negative TNBC type signatures, as you might expect, luminal and luminal. But there was a subpopulation in these tumors that reflected that. And then conversely, the cluster 2 signature that I alluded to was instead not a mature luminal, but a luminal progenitor type signature in terms of looking like normal epithelial cells. And this was highly related to basal-like or intrinsic basal-like TNBC type signatures. So this diversity was there, but of course, what was most interesting perhaps in the analysis that you can see simply by the way the different cells and clusters are shown in these top bars is that each tumor was not restricted to a single TNBC type. And in fact, most of the tumors, although they might have a predominant expression of one TNBC type, had cell populations with multiple TNBC type signatures. So just one example of that would be here, patient 84, which has a substantial population of cluster 3 cells, which have this luminal sort of character. But the same patient also has a substantial cluster of cluster 2-like cells with a particular basal signature. So again, TNBC type, we can then think about it not as has originally been portrayed as a signature that really characterizes the entire tumor, but perhaps rather the predominant cell type in a mix of different cell types within each triple-negative breast cancer. And this paradigm of different types of gene expression profiles being present in multiple subpopulations within each tumor has also been observed in other tumor subtypes, such as brain tumors and other tumors where gene expression subtypes have been defined. But then the question is, what could we learn about these clusters that was really relevant to the clinical behavior of the tumors? So for this analysis, we wanted to look at a number of signatures that were related to the biological behavior and or clinical outcome of triple-negative breast cancer and breast cancer as a whole. So we interrogated the various clusters for their resemblance or possession of signatures. Number one, related to a prognosis signature, which is actually a clinically used signature that was defined to determine prognosis across all of breast cancer, not just triple-negative breast cancer. We then looked at a completely different signature called a metastatic burden signature, which was derived by Zena Werb's group by looking at patient-derived xenograft models and how much metastatic cells these tumors would essentially throw off in the PDX models, metastatic burden, in other words. And then finally, another completely different signature called residual tumor signature, which in fact came from looking at the genes expressed in those cells in the post-treatment tumor of a woman who received preoperative chemotherapy for her primary breast cancer. What were the genes expressed? And the notable finding of this analysis was that even though these three signatures, as I mentioned, their derivation quite different, none of these signatures had a single gene overlapping, and yet each of these signatures was most highly enriched in this cluster 2 subpopulation of cells, which was present in all of the patients that we analyzed in the single cell analysis study, pointing to cluster 2 as potentially a subpopulation of cells that may be driving the bad prognosis of these patients. And so to test that hypothesis directly, we then derived a cluster 2 signature and looked at its ability to predict prognosis relative to these signatures and other signatures for triple-negative breast cancer. And part of that analysis is summarized here. And this is outcome studies derived from bulk RNA-seq data of these triple-negative tumors. And if you apply any of these various prognosis or biological behavior signatures to triple-negative breast cancer, they have absolutely no prognostic ability at all. In contrast, if you apply the now new cluster 2 defining gene expression signature, we believe that by applying this signature to a bulk population, what we're essentially doing is looking at the proportion of cells within those triple-negative breast cancers that possess cluster 2. For every quartile of cluster 2 expression increasing, the prognosis of the patients gets progressively worse. So consistent with the idea that the cluster 2 population is driving the outcomes and giving us actually a clearer view of prognosis than we could get from really any of the existing currently commonly used gene expression signatures that subtype TNBC. So looking at subpopulations can provide clarity as to outcome. So of course, the real challenge then is to identify what are the biological properties of cluster 2 that drive this poor outcome. And I'll have more to say about that, but just to finish this short story, this gives us a window into the value of beginning to look at the subclonal populations. But what we really want to know is what's driving the bad outcome. And so that then leads into the second story, where it's really a story about what are chemofactory triple-negative breast cancers and what therapies might they be susceptible to. And this is a study which started in a very translational way with a clinical trial that we designed many years ago that was led by Steve Isakoff, one of the breast cancer clinical researchers in our program. And this was at a time we were looking for new options for triple-negative breast cancer, and platinum chemotherapy, which had long been used for colon cancer and lung cancer, was virtually not used at all for breast cancer, but it was thought that perhaps for some tumors, it might be a valuable therapy. So this was a very simple clinical trial. Patients who had developed metastatic triple-negative breast cancer, who had received virtually no other therapy for their triple-negative breast cancer, were simply given single-agent platinum chemotherapy, and then we assessed the response and the duration of response. The real value of the trial, in addition to defining subsets of patients, was a lot of the translational work associated with identifying biological characteristics of patients who either responded or didn't respond. And I'll summarize the published data from this clinical trial by saying that our initial hypotheses about the patients who would respond to this therapy were validated. And that was specifically that BRCA2 mutant patients, here shown as BRCA1/2 positives, had a substantial response rate. Over 50% is a very high response rate for triple-negative breast cancer in the metastatic setting. Whereas in contrast, patients who were known not to have BRCA2 mutations in the tumor had much, much lower response rates to platinum therapy. We then went on to collaborate to identify a genomic signature that might infer a homologous recombination defect in patients who did not possess germline mutations, but might have other pathway defects leading to this homologous recombination defect, or so-called HRD. And so this was an analysis of various genomic properties that are associated in tumors with homologous recombination deficiency. I won't go into all of the different patterns of genomic rearrangement and mutation that we're looking at that make up this signature, but suffice it to say that this is actually now a clinically applied signature, which is termed HRD, which is being used particularly in ovarian cancer, and will probably be in the future used in other settings to direct patients for PARP inhibitor treatment. But what we showed in this early study was that, like the response of BRCA2 mutation carriers, patients who lacked mutations but had this HRD signature, here shown in the higher level of the HRD score, were significantly more likely to have complete or partial responses than patients who lacked HR deficiency signature. So all consistent with the pre-specified hypotheses of the study. But after this, of course, what we really wanted to look at were the patients who were at the other end of the spectrum, the patients who didn't respond to therapy. And so shown here is what we call the waterfall plot for this clinical trial. These were the patients who we were able to do genome-wide transcriptome profiling of the patients. The bars tell you what happened in terms of response of the tumor in response to therapy. So the bar goes down, the tumor shrunk. If the bar goes up, the tumor was growing. And there's a clinical threshold for something called de novo progression. In other words, progression almost like the patient's not receiving any therapy, and that's more than 20% growth of the tumor. And so we had 7 patients in that category. So we did the gene expression profiling, and then in an unsupervised way, we said, can we define a signature that identifies the patients who had de novo progression versus those who had all other types of responses to the single agent platinum chemotherapy? And indeed, we were able to do so. And this was work we did a couple of years ago with Shriya Ramaswamy, Ken Ross, and Lei He in the lab. And this was a signature of over 100 genes that were substantially downregulated in the subpopulation of patients who really seemed like the tumors had no response whatsoever to what is otherwise typically a fairly aggressive and effective chemotherapy. But of course, the question was, what is the signature and what it tells us? So the most informative thing we were able to do with this signature was then apply it to a pharmacogenomic database where you had a whole variety of tumor cell lines, not just triple-negative breast cancer, that had been treated with 100 or more targeted therapy drugs, as well as chemotherapy, that had had transcriptome-wide gene expression profiling. And so we could ask, is there any drug that these patients who are so chemo-refractory that possess this highly refractory tumor or HRT signature, are there indeed any drugs that tumors of this feature do respond to? And in fact, there was one clear and unambiguous signature. In fact, the tumors that had the HRT, tumor cells that had HRT signature were in fact highly sensitive to inhibitors of MEK1/2 kinase. So MEK1/2 inhibitors in patients with the HRT signature, the tumor cells seem to be quite sensitive. And you can see that in terms of false discovery of the enrichment for multiple MEK and ERK inhibitors in the HRT positive tumor cells. The second biggest hit was EGFR inhibitors in HRT positive tumor cells, and that will become important later in the talk. So an interesting observation, but what on earth does it mean? What's the basis of it? One of the first questions is, when you think MEK-ERK, you think RAS, and you think of RAS mutation. Is it possible that in all these cell lines that this was somehow a surrogate of RAS mutation? We didn't think so, because RAS mutation is uncommon in triple-negative breast cancer. And indeed, the signature was actually also able to predict response to MEK1/2 inhibitors, even in RAS mutant tumors. So RAS mutant tumors, the line is shifted down in the IC50 for a MEK inhibitor, so RAS mutant tumors are more sensitive. But the predictive power of the signature in mutant tumors was similar to that in wild-type tumors. So the signature is not simply a surrogate of RAS mutation. Still, we were unsatisfied. We didn't know what the signature corresponded to biologically, so we needed to develop an ex vivo model to understand it. So for many years, platinum resistance models have been used and have provided informative information. And so we generated an ex vivo in vitro cell-based platinum resistance model where you can take cell lines and treat them with platinum in a variety of ways to generate isogenic cells with cisplatin resistance. And so we did this for triple-negative breast cancer. You can see you treat them with low doses for a period of time, and they become stably platinum resistant. The IC50 curve shifts to the right. But what was gratifying was this appeared to be a good model potentially to study the MEK inhibitor sensitivity, because in this very model, when the cells acquired platinum resistance, they also acquired MEK inhibitor sensitivity. So the MEK inhibitor curve shifts to the left as the platinum sensitivity curve shifts to the right. And even better, we were able to show that in this type of model, when they got platinum resistant and MEK inhibitor sensitive, they also acquired the HRT signature. So all suggesting this could be a valid model in which to understand the biology of what's going on. So then we carry out a proteomic analysis using one of the commonly used and highly validated reverse phase protein arrays. And there are a number of interesting findings, but among the most informative was the fact that in these cells with resistance compared to the isogenic platinum sensitive cells, there seemed to be a substantial activation of the transcriptional cofactor YAP. So what you can see here is YAP S127, which is on the array, of course, is an inhibitory phosphorylation of YAP, and this is dramatically decreased in the platinum-resistant state. If you do the ratio of the YAP over the inhibitory YAP, you can see that this is associated with substantial activation of YAP in the platinum-resistant state, and this is validated by Western blot of YAP activation. So this was an exciting finding because it went along with a lot of emerging data on YAP in breast cancer and triple-negative breast cancer. And indeed, YAP itself has been associated with chemo resistance in breast cancer, as well as a number of other features associated with poor prognosis in breast cancers. But of course, as a transcriptional cofactor, YAP is likely to regulate a whole series of different gene expression programs. So finding YAP itself really didn't give us much of a specific handle on mechanism and would be a bit hard to sort out. And indeed, it took us a while to figure out what really was the key target of YAP that was conferring the effects that we wanted to study. And that work was done, and it really took the entrance of a new postdoc on the scene, Xiang Ko, who came in and tackled the idea of how can we deconvolute what YAP is doing in these tumors to confer this phenotype. And what he did was a convergent analysis entirely in silico, where he said, what if I look at the genes that are bound by YAP in ChIP-seq, and then I further look at RNA-seq analysis, which he carried out of YAP-regulated genes in triple-negative breast cancer. And then I further superimposed that on genes that were related to the HRT signature. And when he did this analysis for YAP, or for the related cofactor TAZ, a single gene emerged from these convergent analyses. And that was a gene known as RASL2, which is a RAS gap, that is a RAS inactivating factor. And so RASL2, there was some information that it might be an important breast cancer gene, so it seemed like a plausible candidate. Xiang went on to show that indeed YAP is a directly transcriptionally regulated by YAP. And you can see that here where Xiang did CHIP analysis. There was also ChIP-seq analysis to validate the direct association with YAP with RASL2, and then knockdown overexpression studies showing that indeed YAP drives expression of RASL2 in triple-negative breast cancer cells. But does it really add up in terms of the phenotype going back to our original clinical trial? And the answer was yes, and that data is shown here, where if you look at, here we've divided, this is the platinum chemotherapy trial that I described to you, and if you divide patients up simply to those who had the HRT signature versus those that did not have the HRT signature, you can see that they have both increased YAP. This is an activation signature that we derived from RNA-seq in triple negative. YAP is activated, and indeed, so is RASL2 expression, specifically in the HRT subpopulation of tumors. So that's one piece of data, but if you look in the larger world of triple-negative breast cancer, is RASL2 really associated with chemo, poor chemo response and associated bad outcomes? So here, Xiang didn't just do your standard, let's look at the prognosis, better or worse, but he said, I really want to know if this is a chemotherapy resistance gene. And that's the analysis shown here in the upper right, where he looked at the Metabrick TNBC cohort. And he showed that if you look at all triple-negative breast cancer, or triple-negative breast cancer that was not treated with chemotherapy, RASL2 is not very predictive. But if you look specifically at the subpopulation of patients who were treated with chemotherapy, then you see significant prediction where high RASL2 expression is associated with bad response, suggesting that the bad prognosis is specifically related to the chemotherapy response of these patients. And then similarly, if he did a very simple analysis, again, go back to that clinical trial, just divide patients by RASL2 expression at the median, right? So lower than median, higher than median RASL2 expression in the platinum chemotherapy trial. And what you see is that every patient who had de novo progression had high RASL2, and conversely, no patients with below median RASL2 expression had progression. All of those patients either achieved stable disease or a complete or partial response with platinum chemotherapy, all supporting the idea that RASL2 is related to chemotherapy resistance in triple-negative breast cancer. And as you might surmise, RASL2 is not specifically related to the other TNBC type triple-negative signatures that we talked about previously. So RASL2 expression is independent of the known triple-negative breast cancer subtypes, as was the HRT signature that I talked about before. But of course, then we wanted to ask the question, since I've told you about subpopulations and I've told you that we think that we've identified this cluster 2 poor subpopulation that was defined here. Here's again our t-SNE of the single cell analysis with the cluster 2 that gives bad outcome. So then Xiang asked a simple question, what does RASL2 look like in cluster 2? And gratifyingly, we could see that RASL2 expression here shown in pink and purple was highest by far in the cluster 2 subpopulation, and that's summarized by here, where statistically RASL2 is higher in the poor prognosis subpopulation within triple-negative breast cancer. But of course, everything that I've shown you so far is correlative data, so the next step was naturally to show that RASL2 in fact conferred chemosensitivity and chemoresistance, and that's what's shown here. So by overexpressing RASL2 in triple-negative breast cancer cells that have low RASL2, you can confer chemoresistance not only to cisplatin, but doxorubicin and gemcitabine, 2 commonly used chemotherapies in triple-negative breast cancer. And Xiang is still working on the precise mechanism of the chemoresistance, but he knows that it's likely related to the RAS-GAP activity of RASL2, because here you can see that both for chemosensitivity and for PARP inhibitor resistance that is mediated by RASL2, that resistance is only conferred in wild-type RAS tumors, but not when RASL2 is expressed in tumors which already have mutant-activated RAS, suggesting that the ability to modulate RAS activity by RASL2 is important for its ability to confer multi-agent chemotherapy resistance as well as PARP inhibitor resistance. But of course, what we really want is to talk about sensitivity. So the big question was, is RASL2 associated with the entire program with the MEK inhibitor sensitivity? Of course, I wouldn't set it up this way if the answer wasn't yes. So indeed, RASL2, when it's expressed in triple-negative breast cancer cells in inducing chemoresistance, also confers substantial sensitivity to MEK inhibitors. And that's shown here, looking at multiple TNBC lines, both in short-term viability and in clonogenic assays. RASL2 is sufficient to confer sensitivity to this pathway. So how might that work? So surprisingly, so these are studies of time courses of MEK inhibitor in triple-negative breast cancer. And the first surprise was that here on the left is vector, in each case on the left, and RASL2 expression on the right. The big surprise initially was that when you express RASL2, which is a RAS gap and inhibitor, the basal activity, as assessed by ERK phosphorylation, for example, really doesn't change, and if anything, might be slightly higher. When you inhibit MEK acutely, of course, you see decreased ERK, and that's not changed. But what was changed in the setting of RASL2 was actually the feedback activation, particularly in the PI3 kinase pathway. So this is an experiment where Xiang treats with the MEK inhibitor, then washes out and looks at feedback reactivation of PI3 kinase, which is quite strong here under normal circumstances, but is almost completely abolished in the setting of RASL2. And the same thing is observed if you simply give a single dose of the MEK inhibitor and then wait for the feedback activation to occur. It's in fact really abolished when you overexpress RASL2. So we surmise that this might be related to multiple feedback pathways that govern RAS activity once downstream pathways are inhibited. And the clue to which one was most important came when we looked at the correlation between RASL2 expression and these feedback regulators in actual triple-negative breast cancers in vivo. So it turned out that tumors that express high levels of RASL2 actually express significantly lower levels of the Sprouty genes, particularly Sprouty2. High RASL2 is associated with Sprouty, which are negative regulators of RAS. So is this playing out molecularly in our in vitro models? Yes. When you overexpress RASL2, you can see that the Sprouty genes are downregulated, and that is associated with the failure of the rebound activation of particularly PI3 kinase signaling when RASL2 is, when MEK is inhibited. So basal RAS activity is not changed, but the feedback pathway is deregulated. So I won't go through all the ways that we've validated and tested this pathway, but we'll summarize the basics of it here by showing that what we believe is happening is that, of course, RAS activity is important for these tumors. When RASL2 is expressed and RAS activity is inhibited, the tumors compensate by downregulating Sprouty. That's why in primary triple negative, high RASL2 is associated with lower Sprouty levels. Now, in the setting of a MEK inhibitor, normally a triple negative breast cancer cell would compensate by downregulating Sprouty to restore basal RAS activity. But in the setting of RASL2 expression, where Sprouty is already down, it cannot be further downregulated in the setting of MEK inhibitor, and that disables the feedback activation of PI3 kinase, which we believe is at the heart of the sensitivity of these cells to the downstream MEK inhibition. Further evidence for this model is here when he does Xiang does an active RAS pull-down assay. Here you can see treating with a MEK inhibitor leads to rebound activation of RAS, and that rebound activation is disabled in the setting of RASL2 expression. But in real life, we didn't think that this was the entire story. And that was because when we looked at what were the strongest correlations of players in this pathway in triple-negative breast cancer, the strongest correlation with RASL2 expression was actually the EGFR correlation. So RASL2 high expression was strongly correlated with EGFR, which is thought to be a potentially plausible target in some triple-negative breast cancers. And that's true, the strong correlation both in breast cancer cell lines and in TCGA triple-negative breast cancer. And so the model for all of that signaling is that in the setting of high RASL2, which confers chemoresistance and gives a selective advantage to the tumors, Sprouty is downregulated and EGFR is upregulated to maintain RAS activity. So that suggests that two key vulnerabilities of the tumors would be to MEK and EGFR inhibition. So Xiang then went on to test the combination therapy in the setting of in vitro and in patient-derived models. So here you can see a synergy analysis, which really bore out Xiang's hypothesis that these treatments, MEK and EGFR inhibition in the setting of high RASL2 selectively, would be highly synergistic. And these synergy plots, up is synergy, down is negative synergy. You can see that in cells which don't express high levels of RASL2, there's a none or negative synergy with the combination of MEK and EGFR inhibitors. But in the setting of RASL2, you get strong synergy. And this is borne out in these tumor cells by a dramatic potentiation of apoptosis, shown here by caspase-3 activation, which you can see in the staining here when you combine EGFR and MEK inhibition. So of course, we want to know whether that occurs in vivo. These are xenograft experiments that Xiang did. So each of these inhibitors alone, MEK is single agent, EGFR is single agent. I will tell you parenthetically that these have been tried as single agents in triple-negative breast cancer without very much success, which perfectly replicates what we saw as single agents in this study. The single agent, either with or without RASL2, was only very modestly effective. However, when you combine MEK plus EGFR inhibitors, specifically in the context of RASL2 expressing tumors, but not RASL2 low tumors, you get this substantial tumor regression, which is shown here in a waterfall plot for this animal experiment. So this is really great from the point of view of cell lines, but what about in real triple-negative tumors? Xiang then went on to develop patient-derived models, two of which are shown here. These are patients who receive preoperative therapy the way that I described it to you. And in both cases, the patients had very poor responses to chemotherapy. Tumor was collected post treatment that was residual chemorefractory tumor. Unfortunately, both of these patients went on to develop metastatic disease consistent with the chemorefractory nature. The tumor cells were propagated as we predicted. They showed high levels of RASL2 expression, which you can see here in the staining together with EpCAM staining of the tumor cells. And indeed, as predicted, there was potent synergy, but not very strong single agent activity with the combination in these patient-derived models in vitro. So modest activity of each inhibitor by itself, but substantial synergy leading to potent apoptosis in both models with the combination. So fairly good validation for the idea that in patient-derived models, high RASL2 is truly going to be responsive. We'd like to then test this in vivo patient-derived models. And so Xiang looked through the catalog of PDX tumors that he might test to validate his hypotheses, and he identified a good pair of PDX models to test. And so this is basically the chart of chemoresponsiveness to RASL2 expression in a series of PDX models, each shown by a dot. And so he selected one model that was highly chemoresponsive, which had low RASL2 as predicted, and another one that was very poorly responsive to chemotherapy, which, as we predicted, had high RASL2 expression. And this is the chemoresponsiveness. You can see here, again, as predicted, high RASL2 tumor, essentially no benefit from platinum chemotherapy. And what Xiang showed then was that, as predicted, the combination of the MEK and the EGFR inhibitor, in this case of this entirely chemorefractory tumor, was quite potent in inducing a tumor regression and long-term tumor regression in the setting of this model. So quite satisfying from the point of view of all the ways that we can validate this. And we think we have some understanding about what the nature of the signaling is, why it works this way, and indeed how we might identify patients for such a treatment. So, to conclude the real data part of the presentation, I just thought I would give you a schematic about how, first of all, how the pathway works, and then secondly, how we might apply it. So to summarize the RASL2 story, we believe that in RASL low TNBC, obviously RAS activity is high, Sprouty functions as a tonic negative regulator, and EGFR is modestly expressed. In the setting of high RASL2, Sprouty is downregulated to compensate for RAS inhibition, and EGFR is upregulated, and that leads to a specific vulnerability because RAS cannot be further feedback activated once both MEK and EGFR is inhibited, leading to the synergistic sensitivity and apoptosis. So how might we apply this? One possible scenario comes back to the scenario I described to you initially of patients with primary TNBC. Basically, the vast majority of patients with primary TNBC are always going to receive preoperative chemotherapy. We know that those patients who achieve pathologic complete response likely don't need further therapy, and indeed these patients are simply observed. But those with post-surgery residual TNBC require additional therapy and are being challenged actually in the field now is to decide what therapy is right for which patient with post-surgery residual TNBC. And I would propose that one possibility is that patients in this category with low normal RASL2 may have still some intrinsic sensitivity and could benefit from additional chemotherapy. Studies have already shown that in this setting, if you give additional chemotherapy, a subset of patients, but not all, do benefit. But that those patients who have high RASL2 may be unlikely to benefit from additional chemotherapy, but might be better candidates for such therapy, but also including combined MEK and EGFR inhibition to kill off this subpopulation of cells which really is not responding to the therapy the patient had and might be ultimately driving poor outcome, metastasis, and ultimately failure of treatment. So to summarize what I've told you, TNBC is a disease characterized by extensive inter- and intratumoral heterogeneity, and these are both important for outcome and drug sensitivity. Chemorefractory tumors may have an unanticipated deregulation of feedback adaptive signaling in the RAS-mediated pathways that confers a specific therapeutic vulnerability, and that RASL2 might be one way to identify tumors that have this hallmark because it's sufficient to confer sensitivity to MAP targeting agents and combined MAPK EGFR inhibition that we might be able to employ in this selected subpopulation of tumors to improve outcomes. In the last minute, I'd like to acknowledge everyone, particularly Xiang, who led all the RASL2 work, essentially, Miraban, who led all the single-cell data analysis, and so many collaborators in computational biology, in the MGH breast cancer program, in medical oncology, surgical oncology, MGH pathology, to all of our core facilities, and particularly all of the funding agents who supported our work over many years. Thanks very much for your attention. I'm happy to take any questions.
Thanks very much, Leif. This was a great presentation. I'm going to give a few minutes of time for people to enter their questions in the Q&A box, and then we'll call on them. So feel free to type away. Let me start with one question. You started off by talking about the subset of triple-negative breast cancers with HRT defects versus those without. Can you just comment the RASL2 phenotype that you've described? Is that particularly relevant to HRT positive or negative? And then you had described some differences between genetic and epigenetic inactivation in African-American tumors versus those in Caucasian patients. Any sensitivity or differences that you could tell there?
Yeah, excellent question. So for the first question, I think we think of RASL2 high TNBC as kind of the anti-HRD. HRD tumors have general chemosensitivity. Some of them with high HRD, high deficiency, are PARP inhibitor sensitive, and those are the ones who are going to benefit most from both standard therapy and standard PARP inhibitor therapy. RASL2 is the exact opposite end of the spectrum. The ones that are going to be the most chemo-refractory don't have HRD, and in addition have this particularly chemo-refractory property that really limits their benefit from any type of chemotherapy. So really the opposite of the HRD tumors. With regarding the genetic versus epigenetic activation, I mean, this is a very active area of interest for ours simply because there have been no clinical trials looking at epigenetic inactivation and its correlation with PARP inhibitors. What we know is that in the study that we did with Gad Getz, that those tumors with the epigenetic inactivation have quite strong HRD signatures. And we know, based on some clinical data, that other types of tumors, such as PALB2 mutant tumors that have these types of HRD signatures, even though they're not BRCA1 mutant, do respond to PARP inhibitors. So we really think that there's some ability to address an unmet need here by, in a systematic way, identifying these patients. We think that this is really something we need to go after clinically.
Thanks, Leif. So let me go to the Q&A box. Our first question is from Dario Marchetti. Let me read this. Have you translated the TNBC4 type signatures in circulating tumor cells from the same patients?
Yeah, it's a very good question. And I think that, TNBC type, because it was derived from bulk signature, reflects both the tumor epithelial and non-tumor components. So, we applied TNBC type, as a way to kind of understand various properties, but their application to the single cell analysis is relatively limited based on the fact that they were derived from bulk tumor analysis. So, the amount that we might be able to learn, the true answer is we haven't done it, but it's possible that the amount that we can learn might be somewhat limited given the fact that those are really bulk derived signatures and have more, and have a lot to do with other elements of the tumor, not just the single cell. So limited application to applying to single cells, but certainly potentially worthwhile to correlate with subpopulations within the tumor cells. Is cluster 2 is cluster 2 signature in the circulating tumor cells, and that's something that we would like to pursue as well.
So then we have two questions from Shamala Maheswaran. The first one is, did the HRT refractory patients have a prevalence of bone metastases over visceral mets?
So by and large, triple negative tends to have more visceral and less bone than hormone receptor positive. And the HRT seemed to be, as far as I recall, no exception to that rule. In other words, it was predominantly visceral, not bone. But it's an interesting thing to look into based on the unique biology of those HRT tumors. I don't recall a difference. I'm not sure. I'll have to look at that again, but it's an excellent question.
And then her second question is about the RASL2 phenotype. Is it associated with regulation of mTOR PI3 kinase activity, or is it just PI3 kinase pathway in general?
Yes, so that's really the great question about, what is downstream of this pathway deregulation that is related to the phenotype, particularly of chemoresistance. So we think that there's likely to be an mTOR component that is downstream of that signaling deregulation that is associated with the chemoresistance. And indeed, that is the work that Xiang is doing now to figure out how that pathway is working. And we think that it probably has something to do with DNA damage checkpoint signaling that intersects with PI3 kinase and mTOR. But indeed, that's exactly the direction we're going.
And if I can add to that, I would have expected that rebound AKT signaling would confer anti-apoptotic properties and would be a bad outcome, if you will. Your data suggests that the tumors that don't have rebound AKT upregulation actually do worse. Were you surprised by that?
So yeah, it's very interesting because I think that, looking at MEK inhibitor is a very specific way to perturb the system, right? And so, that's really what accounts for this duality, that highly chemorefractory, but really sensitive to specific ways of targeting growth factor. So what we think is that the compensating mechanisms really are the things that probably confer worse outcomes until you perturb specifically those pathways as a way to leverage the adaptations the cell had to make to upregulate those compensating pathways. In other words, high EGFR, which is selected for based on the deregulation of the signaling, probably does a lot of bad things to promoting bad outcomes in the tumors. And it's only if you know how to perturb it in the right way that you can then exploit that as a vulnerability, because clearly it's associated with bad outcomes in response to chemo.
So since we're getting short on time, I'm going to summarize the next three questions, which really talk about the diagnostic tools that you can to pre-identify RASL2 positive tumors. Are there any clinical or other correlates that would identify those within TNBCs? And then if you look to the future, how would you devise a therapy? Would you treat with EGFR and MEK inhibitors upfront in TNBC or after progression on taxanes?
Yes, again, excellent questions. And I think that, because RASL2 is an individual gene, and the studies that we're doing now, this is exactly along the line of what we're thinking. The studies that we're doing now, we're trying to define whether there are thresholds which really dictate the phenotype that we're seeing, right? That will correspond to our various models from a point of view of molecular diagnostic standpoint. Are there either thresholds, or in addition, what we have found is that there are other companion diagnostics which go along with deregulation of the signaling pathway. Of course, there are lots of other things related to the deregulation that I wasn't able to go into that might, as a multi-gene diagnostic, be better at picking out the specific phenotype of the tumors. And then regarding the therapy, I provided one example of how one might use this, and particularly in the setting of patients who don't respond to chemotherapy. Because, everybody believes triple negative should be treated with chemotherapy. Most patients with triple negative are going to get chemotherapy. So the idea is that we would target this therapy to specific populations which express the biomarker and that might be demonstrated to have some degree of chemorefractoriness. And that's somewhat of a practical consideration in the way clinical trial development is done. You look at tumors that don't respond. I mean, the good news here is that we believe that by selecting for tumors that don't respond, you actually are further selecting for the phenotype that's going to be associated with the sensitivity to the targeted inhibitor combination. So in this way, doing that kind of selection that's necessary for trials is actually going to select for the patients who are going to benefit most from the alternative approach.
And there's another question from Nirmal, which is RASL2 upregulated after exposure after exposure to chemotherapy? Have you looked at that?
I'm trying to think where we've looked at that. I think the answer is yes, and the question is whether it's upregulated on a per cell basis or whether instead it's upregulated as a result of selection for the RASL2 high subpopulation that doesn't respond to chemotherapy. I'm guessing that perhaps at least part of it would be the latter, that is, upregulated in the tumor as a whole based on selection for the subpopulation that's high, being chemorefractory.
We have a question from Anne asking for the mechanisms by which RASL2 overexpression mediates specifically the sensitivity to PARP inhibitor or cisplatin resistance. So both mechanistic and specificity questions.
Yes, exactly. And I think, those are the studies that are ongoing. I mean, we do believe that it is related to the deregulation of growth factor signaling that occurs, and that there is a crosstalk, which has been reported by others, with the DNA damage and repair checkpoints, that the crosstalk is perturbed by that, leading to alternative signaling, which changes the checkpoint and repair mechanisms that occur when DNA damage occurs, and that it might be in response to multiple types of DNA damage caused by PARP, damage caused by platinum, damage caused by a topoisomerase II inhibitor like doxorubicin, that accounts for the kind of pleiotropic DNA damage resistance that we see in the RASL2 high tumors. But these are the things that we're working on now and trying to get a clear mechanistic handle on this crosstalk.
And Leif, have you looked at other subtypes of breast cancer or other cancers? Do you see any effect or any impact of RASL2?
Yes, yes, which I didn't mention, which is a whole nother field. It turns out that in HR positive breast cancer, RASL2 is actually a favorable prognostic factor. So in this, and probably has to do with something to do with the crosstalk between RASL2 and growth factors that impede on estrogen signaling. And so interestingly, it actually seems to be behaving the opposite way in hormone receptor positive, again, probably having to do with crosstalk with estrogen signaling in that context.
And outside of breast cancer, have you looked?
Not so much. I think that based on our initial cell line screens, we would predict that RASL2 would have some ability, but the context might be different. For example, in terms of, maybe it wouldn't be EGFR that would be upregulated, but another factor that might be upregulated, or different ways that signaling is compensated for when RASL2 is overexpressed. We believe that by and large, it may not have a big role in tumors that have a high prevalence of RAS mutation, because, for example, as Xiang showed, that in the setting of RAS mutant tumors, RASL2 does not confer chemo resistance, again, consistent with the idea that it has to do with the signaling pathway and RAS deregulation that's associated with the chemo resistance.
Great. We're both out of time and out of questions, so I want to congratulate you on great work and for an excellent talk.
Thank you very much. It's nice to see everybody. Thanks for your participation.